Okay, welcome everyone. I'd like to begin by acknowledging the traditional owners of the land where which the Institute is built, the Wurundjeri people of the Kulin Nation, and pay my respects to the elders past and present, and extend that respect to any other elders from any other Indigenous communities that may be present. So today's talk, as you can see, is by Rhys Allan from Immunology Division, and this is part of his lab head renewal process. As a way of background, Rhys performed a spectacularly successful PhD at the Micro and Immunology Department at the University of Melbourne, followed by an equally successful postdoc at the Institut Curie in Paris. And it's during this postdoc in Paris where Rhys got interested in understanding how genes are regulated in the immune system, particularly at an epigenetic level. So I was delighted in 2011, I believe it was, when Rhys decided to come back to Australia and join my lab and continue on his work on gene regulation in the immune system, something that's dear to my heart. And Rhys, as you'll see, has been spectacularly successful. He rapidly, upon coming back to Australia, was awarded a prestigious ARC Future Fellowship and then appointed a lab head in 2015. And I think, and he used his time as lab head to greatly expand his sort of interests in the area of gene regulation. And hopefully you'll see from the presentation, I think that he's done this in a very innovative way. He has used some really cutting edge genomics techniques. And I think most importantly, he's tackled the question of how the DNA works within our nuclei at multiple levels of scale. And I think that's a really important way of thinking about how the whole system is set up in totality. This sort of work has led to some really fundamental insights in how the immune system works, as you'll see today. It's also led to many successful collaborations with Rhys, and so he's really embedded himself very nicely within the Institute. And also, very excitingly, has led to some translational opportunities, which I think you also mentioned. So please join me in welcoming Rhys to the podium to present his seminar entitled Deciphering the Epigenetic Code in the Immune System. Thank you.
Thank you very much, Steve, for that lovely introduction. So I wanted to start with a bit more relaxed approach, just to give a historical overview of our lab. So we started in 2015, and it was with myself here, Dane and Nadia. We've been reasonably productive. So with Dane, we published a few papers. Then Tim appeared, the bigger version of Tim, and Hannah as well around this time, and we published a few papers together. So we've been quite productive there. Then Luke, and a lot of the work we're going to talk about today is Luke's work. And then Christine as well, and we published recently as well. But one of the things that really defines our lab actually is a different aspect, which is our production of babies. So we've had 6 babies in the last 4.5 years. So that's been pretty full on, and maybe you'll understand if I start falling asleep towards the end of my seminar. Well, of particular note is this cluster here, which has caused us a lot of very interesting lab meetings where lots of thoughts have been forgotten, or probably brilliant ideas have been lost, actually. So what my lab is really interested in is what many labs are interested in here, is the development of the immune system. And we're really interested in understanding how cells from a stem cell become committed to their individual lineages, which we know are functionally distinct. So what occurs is you have this major transcription factors have been defined. Cells differentiate down these specific pathways, become committed to their lineage. And for example, you have the adaptive immune system. T cells and B cells are important for killing, helping, making antibody, and also forming long-lived memory, whereas innate cells like these monocytes and granulocytes are really important for early responses, and they're functionally distinct from these other cell types. And what we know is that these cell types have their own unique transcriptional profile that define the phenotype and function of these cells. And so really the interest of my lab is to understand what actually regulates the lineage-specific transcriptional programs or signatures that define the function of these cells. And so in a simple sort of way, what we start with in terms of gene regulation is the DNA sequence itself. So the regulatory regions, such as promoters, which lie around the transcription start site of a gene, and enhancers, which can be 10 to hundreds of kilobases away from their target gene promoter. The sequences in there dictate the gene expression. And what they allow for is transcription factors to bind to these sites and bind to specific motifs within these regulatory regions and recruit cofactors which can drive transcription or repress transcription. And then there's an additional layer of complexity upon this, which is the epigenetic modifications to the chromatin. For example, we have modifications to histone tails which are associated with active gene transcription, such as acetylation of lysine residues on histone tails, and which is also associated with the euchromatin, which is a more open gene and active region of the genome. Or we have modifications like trimethylation of certain lysine residues, which are associated with heterochromatin or gene-silenced regions. And so basically it's very convenient to talk about the genome as a linear ball on a string in this sort of sense. But what we really know from more recent work is that these enhancers and promoters have to come into physical contact generally to drive transcription of their target gene. And recent work has shown that you can basically form this type of loop where the enhancer and promoter can now get into close contact via a process recently termed loop extrusion. And we know that that process is extremely important because if you have defects in this type of looping, it's been shown that that creates developmental defects, heart disease, and cancer. But there's still really many questions that remain about how the genome is organised during the immune system, which allows us a lot of interesting questions to ask. So basically the aims of my lab is, as Steve suggested, is to understand the transcriptional regulation of the immune system. And what I want to talk about today is the two major aims of the lab. One is about targeting the epigenome, which is a more mature project, which is nearing completion. And the other one is the work that we're still ongoing about how the genome is organised. So what I wanted to do was just to start by talking about this project, about how we're going about trying to target the epigenome to modulate immune responses. And the rationale for this work is that if you can understand how the epigenome modulates the T-cell fate, potentially we can reveal novel targets to modulate the immune system. And what we've specifically focused on is histone-modifying enzymes that are associated with gene silencing. And the reason we did this was because we know there's many small molecule inhibitors of these enzymes which you could potentially then use to modulate immune responses. And we focused on studying allergic asthma, and that's because it's based on my previous work, which was focused on this kind of area. And just briefly, what we actually found in our most recent work is that in a normal situation, cells which are responsible for driving allergic inflammation, which are called T helper 2 cells, they critically rely on the presence of this histone methyltransferase EZH2 to drive the immune infiltration, lung inflammation, and decreased respiratory function that's associated with allergic asthma. And so we use this knowledge then to do experiments where we used a small molecule inhibitor of EZH2, which is actually currently in clinical trials as a potential treatment for cancer, to inhibit EZH2 function. And effectively, when we did this, we could switch off the allergic asthma immune response, which resulted in decreased infiltration of immune cells, decreased inflammation, and increasing of the respiratory function in mouse model systems. And I've highlighted these little stars here because we could also do this in a therapeutic setting. So when the animals had already had established asthma, treatment with the drug could reverse the disease, which is quite unique. And so we published this just recently in JCI Insight. And we also got quite a bit of media. Thank you to our media department. And here's Christine doing an interview with Channel 7. So we're very happy with that. But I was also happy personally because in my local kinder, one of the dads thought I was the resident bum, the bum of the neighbourhood, and then he saw me on TV and he realised that now I'm a very important person in a lab coat. So I made it. He'll never think like that again. Anyway, so what we're going to do in the future is explore the possibility of testing this strategy in the clinic and try and push this work forward. And so now what I'm going to spend the rest of the time talking about is our new work, more recent work on understanding how the genome is organised in the immune system. And so when we started this research, what we realized was that we were basically faced with this conundrum that every nucleus contains around 2 meters of DNA. And so how would you organize this, this chromatin, this DNA, to drive the transcriptional programs that I described earlier? Is it that we have very nicely organized, packaged nuclei in a very non-random way? Or is it just like a big bowl of spaghetti that's all just mushed together and somehow it works its way out? So what we did to try to answer this is we designed a relatively straightforward experiment. We took these T cells, B cells, and granulocytes from mice, so functionally distinct populations of cells, and we assessed the global genome architecture using a technique called chromatin conformation capture. So briefly touch on this technique because we used it basically for the rest of the talk. It's a technique called in situ Hi-C, which was published in 2014. And what this technique involves is affixing the nucleus with formaldehyde, and that effectively cross-links all the interacting chromatin. And then we digest the chromatin with a restriction enzyme, and importantly, after this step, the pieces of interacting DNA are still held together by the cross-link. Then we tag the DNA with biotinylated nucleotides, we ligate it back together, shear and isolate these interacting fragments, and then we perform a sequence, next-generation sequencing, and we map this back to the genome. And we basically create something which we call contact matrices, where so if we're looking at, for example, we're very interested in regions that were interacting, this blue region and this green region here, and if we see interactions, we see intense spots or read pairs in these bins here. And so we did this experiment and to ask, really, is the genome of the immune cells organised or not? And the answer is yes. So in fact, what we found was when we took granulocytes, B cells, or T cells, you could see very distinct differences in their genome organisation using this technique. But critically, this relied on the bioinformatics package that was made by Aaron Lund and Gordon Smythe in our bioinformatics division, which we call Diff Hi-C. And what that enabled us to do was look at the differential interactions between the populations and call statistically different statistical differences. And basically what we found was there was around 20,000 differential interactions between these populations, suggesting they are very distinct in their genome organisation. When we looked at these played plots, or these contact matrices, what you could see, these are made up of very lineage-specific interactions. So for example, here in T cells, this is the BCL11B RNA-seq. You can see it's very highly expressed in T cells and not in these other lineages. And you can see a very nice interaction that's formed between the promoter of BCL11B and a very distant enhancer, which is actually, we know is about 800 kilobases away. And that doesn't exist in B cells. In contrast, if you look in a gene that's expressed highly in B cells and not in the other lineages, you can see very nice interactions with this gene and distant enhancers, and similarly for granulocyte genes. So importantly, what that allows us to do is to then build on this knowledge, and we can, now that we know that there's a distinct genome organisation, we can then look at how we can then basically build and ask lots of interesting questions, and we've been able to ask and answer some of these questions about the principles of genome organisation in the immune system. And what I wanted to do today was to specifically, or to focus on, or not focus on, to give you basically a snapshot of some of these individual projects which we've completed and published, and some of them that are ongoing, and then expand on one of them to give you more of a taste of how we really go about this work. And I wanted to highlight the individual, the different approaches that we're taking to answer the questions. So the first question we wanted to address is, do lineage-specific gene regulatory interactions occur within chromosomes or between chromosomes? And the reason why we ask this question is because there's been this long-standing belief that there's this stable interchromosomal interactions that exist to regulate T-cell differentiation, as this was published in 2005 and very well received by Richard Flavel's group. And what they propose is that they could detect in resting lymphocytes the stable interaction between interferon gamma and the Th2 loci, that as cells then differentiated, these interactions separated out. And Dimitri Koussis wrote this commentary about it in Nature called Kissing Chromosomes. So it sounds very lovely. And so what we decided what we would do is, well, what we were doing basically with our work anyway in the process of it, is to revisiting this with our unbiased Hi-C approach, genome-wide, looking at our mouse and human immune cells. And when we did this, what we found was the expected interactions within chromosomes and between centromeres of immune cells as was expected. So for example, here, this is chromosome 10 in this region, and we can see nice interactions between in the Th2 loci within the chromosome, and here on chromosome 11, we can see nice interactions around the interferon gamma locus in CD4 T cells. However, we couldn't confirm the previous T cell specific interactions. And you can see here Tim made this plot, which is a bit extreme, but basically this is the region you would have expected to see interactions, and we couldn't see them at all. And so, what we think is happening here is that the techniques used in the past were very biased towards finding interactions, and this is a much more unbiased approach. And generally, we very rarely saw gene regulatory interchromosomal interactions existing at all. And therefore, we propose that lineage-specific gene regulation is driven by cis and not transchromosomal interactions, at least in a stable sense. And we published this last year in PLOS Genetics. And so I guess more boringly, we propose that maybe they're celibate chromosomes, that they don't interact. It's a bit sad, I guess. And so the next thing I wanted to talk about was an interesting question about how you actually establish lineage-specific architecture. And the reason why, so we ask how is cell type-specific genome organization established? And the reason we ask this is because the proteins which have been implicated in genome organization and structure are generally ubiquitously expressed. So it's hard to imagine how they would establish lineage-specific architecture. So what we did with Stephen Nutt is we speculated that lineage-specifying transcription factors may play a more direct role in organizing a genome. And that's because they display this lineage-specific expression. And we know that many of these master-regulated transcription factors bind to many sites outside of genes and many sites throughout the genome. And so, for example, here, BCL11B in T cells is known to those thought to play this master regulator role. PAX5, as we've probably heard about many times, and PU1 here in granular sites. So to ask this question, do these factors have a more global role in organising a lineage-committed genome? We focused in specifically on commitment to the B-cell lineage and the role of PAX5. And we know that commitment to the B-cell lineage begins at this pro-B-cell stage just here, and that PAX5 is absolutely essential commitment factor for commitment to this lineage, and which is work that Steve did many years ago now. And so in the presence of PAX5, the cells will progress through B-cell to differentiate into mature B cells that can eventually make antibody. But if you remove PAX5, what happens is you get blocked at this pro-B cell stage, and depending on the different stimulus, cells can actually de-differentiate. And so importantly for our rationale, the PAX5 binds to over 20,000 sites in the genome, many outside of genes. And very useful to us is the PAX5 deficient pro-B cells. They grow extremely well in culture. And so what we did was we used our high-C technique and we compared the genome of a wild-type pro-B cell to the PAX5 deficient progenitor B cells. And what we did was we focused specifically, at least for this example, on PAX5 bound sites. And to begin with, we actually looked at regions that were not bound by PAX5, and we found if you compare the wild type and the knockout, the interactions have an equal likelihood of going up or down. But if we then focused in on sites that were bound by PAX5, what we noticed was that almost exclusively a loss or a weakening of the genome architecture in the knockout PAX5 knockout cells in regions where PAX5 was bound. And importantly for our work, what we were able to do is use a system which Steve had developed where we could induce PAX5 to go back into the PAX5 deficient nucleus. And then what we saw was an increase or a creation of the interactions back again, specifically in these sites which were bound by PAX5. So that was quite definitive, and that allowed us to basically suggest there was a new role for these lineage-specifying transcription factors in organising the genome. And we published this in Nature Immunology last year. And this is a lovely picture made by Atsuko here, which we submitted to the journal as a lovely cover, which was rejected. Which happened to me a lot, actually, submitting papers and getting rejected. I'm used to the rejection. Anyway, so we now know from our own work and from others that this looks like it's a generalizable phenomenon. So we've shown that PU1 in myeloid cells is also critical in a similar way. KG Zhao and Alan Rothenberg's lab showed that BCL11B in T cells plays a similar role. And during reprogramming, the work of Thomas Graf's lab has shown that these reprogramming factors similarly have a role in organising the genome. So now the next thing we wanted to do was we wanted to ask the question whether we could use our Hi-C data sets comparing different cell types to potentially identify novel regulatory regions. And so what we did was basically what we know from our data sets is that there's around 100 interesting regions that look like they're lineage specific and potential enhancers, novel enhancers for T or B cells. I just wanted to highlight one we found, which is around the important T cell gene called GATA3. And what we can see in our Hi-C maps, when you look in CD4 T cells, we can see here's the GATA3 gene just here, and we can see 3 distinct potential enhancers that interact with the promoter of GATA3, but which don't exist at all in B cells. So it looks like a lineage-specific interaction that may regulate GATA3 expression. We call them region 1, 2, and 3. And Luke in the lab has done a huge amount of work using T cell lines which express high levels of GATA3, and he's performed a CRISPR-Cas9 excision of each of these individual regions in cell lines. And what he found was that region 2 was essential for the expression of, or high-level expression of GATA3. So there's a potential enhancer region. But interestingly, this kind of work can reveal other things. So then Luke looked inside this region and we found a very poorly annotated potential non-coding gene, which then he was able to obtain the full sequence of and show that this gene now has very high expression in the thymus, as does GATA3. And more recently, he's shown that there's very nice expression of this novel gene correlating very well with the expression of GATA3 throughout T cell development. So that gives us another interesting thing to study. And from what we're going to do in the future is continue to study the function of this potential regulatory element. And so what I'd also like to talk about is whether we can study the role of large compartments in organising the genome. And for example, what we've decided to do is look at the role of heterochromatin. And we asked, what role does heterochromatin actually play in the global genome organisation? And what we found, what we're lucky to have had in the lab was we have basically a model of heterochromatin loss, where it's when we remove these 2 histomethyltransferases that are responsible for the H3K9 trimethylation that defines heterochromatin. So we would call these double knockout cells. And what this results in is a loss of the nuclear membrane integrity and premature aging of the immune cells. So normally in thymocytes, you see a very nice lamin B staining around the nuclear periphery, and this is basically lost or misshapen in the double knockout cells. So it allows us to ask, how does this actually alter genome organisation? And what we found by doing lamin B ChIP-seq and our Hi-C is that you lose these large domains of chromatin interactions, which define heterochromatin, in fact. So you can see here these large regions that are lost in the knockout cells. And when we looked at these regions, what we found was that we basically lost the interaction of these genomic regions with the nuclear periphery. So these purple in the control cells, purple and blue regions are regions of chromatin that are interacting with the nuclear periphery. And you can see in the double knockouts that's really lost. And what we're doing now is we're using computational methods to try and perform 3-dimensional reconstructions of this. So what we can see here is that you can see these purple and blue regions. I better play the video thing. Okay, here we go. The purple and blue regions that are normally thought to associate with the periphery, we can see they're really lost in the double knockout cells. And I like to try and make this cool picture, but anyway. And so basically what we can say from this is that we've got, this suggests that the heterochromatin plays a very important role in global genome organisation. So now what I wanted to do was I wanted to just expand on one of these aims and to go into a bit more detail about something interesting and something exciting that we've been working on, which is trying to understand a very specific question about how the genome is organised during an immune reaction. And so, of course, we chose to study B cell differentiation into antibody-secreting cells because that is one of the most well-studied models here at WeHi. And for us, it allows us to study genome organisation in the context of activation, cell division, cell cycle, differentiation, and time. And it also fulfills the criteria of me eventually becoming part of this B cell program. Replacing DT, but I mean, this is an old picture of it. So So what we know about B cell differentiation is you start with a quiescent follicular B cell generally. The cells receive a stimulus, they become activated, and over time they then develop into antibody secreting either plasmablasts or plasma cells. And so we've taken a very well characterized model in the lab, which is to take follicular B cells, activate them with LPS, and then study the genome of activated and plasmablast cells. So these are the antibody secreting version of that. And so to go into this in a little bit more detail, you take these resting quiescent follicular B cells, and when you activate them with LPS, they start to blast and increase in size, and then they have a really long delay before their first division, which is around 30 hours or so. And then once they start dividing, they undergo this really rapid division, which is around every 8 to 10 hours. And over time, they differentiate. So the more divisions they undergo, the more likely that they are of differentiating into plasma cells. And so what we did was that we isolated follicular B cells or the plasmablasts or activated B cells 4 days after LPS activation. And we firstly asked how transcriptionally different are these cell types. And we found, as probably expected, that there's very large changes in transcriptional. And we can see these are the differentially expressed genes here in the thousands of genes that are different between each of these populations. And also, as expected, if we look at specific genes, we found increased transcription of genes such as the gene that encodes BLIMP1, which is PRDM1, IRF4, and another gene which is called EBF1, which we know goes down over time as cells differentiate into plasma cells. So now we wanted to ask, does chromosome architecture change during this B cell differentiation program? And so we went back to our Hi-C and we performed Hi-C on these samples. But in this case, what we've done now is we've used a new package, an EdgeR-based pipeline that's been created by Hannah Coughlin and Gordon Smythe, which now allows us to get gene-level information from our chromosome organisation data. And the way that this is done is basically what we do is we count the number of interactions with every promoter in the genome, with every promoter or gene promoter in the genome, which is effectively taking computationally 24,000 probes, and giving each gene a score based on the number of interactions that occur with the promoter from distant sites. And when we give each gene a score, we can then do a classic Gordon-Smythe analysis of looking at the differential interactions with promoters. So we were able to analyse all the statistically significant differences in interactions between the populations. And when this is just an example of what kind of data we get when we use this analysis. We can recapitulate our TB and granulocyte data as we did in our early differential interaction analysis. But we can also see that we now get thousands of genes that have differential interactions around their promoter when we compare B cells to CD4s or B cells to CD8s, but not when we compare CD4 to CD8, we get very few differences as we have shown before and as expected. And what's very nice about this type of analysis is that we can also get this specific gene information. So here's another example of this B-cell11B gene again, and you can see that there's very nice interactions existing in T cells. And we saw with CD4 and CD8 T cells, we get a very nice number of, nice score, I guess, or counts around this B-cell11B gene in CD4 and CD8 T cells, but not in B cells as expected. So now that that's working, what we did then is we asked this question, does the genome reorganise during B-cell differentiation? And what we found was that yes, indeed, there are large-scale changes in genome organisation between these populations. So there's change between the follicular B cells activated and plasmablast. And these are, once again, thousands of differences in genes which have changes in interactions around their promoters. And the genes are as expected. For example, we know PAX5 goes down-regulated over time as cells differentiate into plasmablasts, and also they have decreased interactions around their promoter, similar for BCL6, and the opposite for genes which we know go up over time. So, for example, BLIMP has increased interactions around its promoter, and similar for IRF4. So the next thing we wanted to do was we wanted to ask, when do these changes actually occur? So it could be that the interactions occur slowly over time, over the cells divide, and then there's a more rapid wave as they differentiate from activated to plasmablast. Or it could be that there's these sort of waves of genome organisational changes that could occur. And so to get at this, what we decided to do is we decided to just look at one time point And we decided to isolate cells just prior to the first division and compare with the others. And the reason why we chose just prior to the first division is because that's thought to be a time when there might be some sort of fate decisions are made or reprogramming prior to the cells entering into cycle. So Luke painstakingly worked out exactly when the cells is about to undergo their first division here, and he used CTV, which we know is diluted as cells divide. And what he showed, what we did was we isolated cells which hadn't divided yet, but it expressed this activation marker CD69, so they had received stimulus and they haven't divided. And then we performed Hi-C on them and we integrated that into our analysis. And what we found was that, in fact, actually, surprisingly, these cells prior to the first division had undergone a large amount of genome reorganization. And interestingly, what occurred was that the genome that was reorganized prior to the first division was almost identical to that of the cells that have divided many times and were taken 4 days later. So you can see here, this is the number of differential interactions around promoters. Between follicular and 33 is a large number of changes. However, between the 33 and activated cells taken 3 days later, they're almost identical. And then there's this other wave of changes as activated cells differentiate into plasmablast. And interestingly, we can pick out genes like BLIMP. Again, you can see actually there's increased interactions around the promoter even prior to the first division, and that basically predicts expression that you might see that we saw later by RNA-seq. So that's actually quite against the expectation. I think in the field, it's thought that the genome will probably undergo this reorganizational event after mitosis. So it's quite surprising as a result. So what we can say now is that it looks like as though there's these 2 waves of genome organizational changes as cells differentiate into plasmablast. Now, the next thing we wanted to do is we wanted to ask, like, how early did these changes occur prior to the first division? And so doing Hi-C is quite expensive, so we didn't want to do too many time points, but we had to pick some. So we basically did, I don't know, some sort of game in the lab. We were trying to pick what the right time points were, and we all had to go. And ultimately, we would look at 3 hours and 10 hours after activation, and look at how the genome changed. And I think actually Gordon Smythe paid for the 10-hour guys, so thanks, Gordon. Well, we did Hi-C again, 3 and 10 hours after activation, with the idea of trying to find out when the changes actually occurred. But unfortunately, or interestingly, what we found was actually even at 3 and 10 hours after activation, there was very few differences or almost no changes in genome organization at this early time, whereas most of the changes we see have occurred by 33 hours. And so as you can see here in this graph, there's almost nothing going on here. And then between 10 and 33 now, there's a big change in architecture. But that's quite interesting as well, because we know from many people's work on our own that if you look at RNA-seq, you can see that most of the differentially expressed genes actually occur, a lot of the differentially expressed genes occur in these first couple of time points between 3 and 10 hours. And so what that seems to suggest is there must be a pre-existing genome architecture that dictates that early expression of genes after LPS activation. And that we have subsequently analysed this kind of, and we find that there is a correlation between chromosome accessibility and transcription that we observed. So now we can go back to our model, and we can say, actually, this looks like the genome reorganises in a more discrete wave here, between 10 and 33 hours. And interestingly, as I just showed, the transcription of large amounts of transcriptional changes appear even prior to this point. So if we revisit our model again, what we can say is that changes that occurred at 33 hours are basically maintained in the activated B cell population as they divide, and then eventually these cells will differentiate into plasmablasts. So I want to go back to this unexpected result that the genome reorganized prior to mitosis. And we wanted to then try and understand at which stage of cell cycle does this actually occur. So we know that when we activate a B cell, they start as a sort of G0 quiescent cell, and then they become activated and they drop into G1, and they spend a really long time in G1 into early and late G1. Eventually, around 30 hours, they just go into S phase, and then they start to go to G2M and divide. And then after that, they spend a very little time in G1, and they're rapidly cycling and spending most of their time in these S and G2M. And so what we're able to do to examine this is to use this Fookie system, which is very well known, and in which the cells which are in G1 are marked with a red fluorescent protein, and cells which are in S or G2M are marked with green. And so what we did was we used mice that express these Fookie constructs. We isolated the B cells, and then what we chose to do was look at this 33-hour time point again, isolate the undivided cells that were activated, and now take the cells that are still in G1, they're still red. And what this allows us to do is to ask, do the changes actually that we saw in genome organization occur prior to DNA synthesis? And what we found when we did this was, indeed, it looked like a large amount of the changes that we saw occurred prior to DNA synthesis. So if you see here in this graph, you can see here that the changes that occurred, we saw large changes in genome organization between 10 and the 33 red time point, and very few differences between 33 red and the 33 total or 33 red or activated B cells, suggesting that these changes all occurred prior to DNA synthesis. So we can revisit this and we can say that basically changes that occurred in this stage prior to entering to DNA synthesis are now maintained in the activated B cell population as they progress on through their divisions. And so we showed that this first wave occurs prior to entering into DNA synthesis or S phase. And we know that the cells undergo rapid division, as the cells undergo rapid division and expansion, the architecture is maintained. And these cells have very short G1. And then eventually these cells will differentiate into plasmablast after 3 or 4 divisions. So what we postulated was that maybe there's a prolonged G1 phase that might be required for a genome reorganization. And so we asked the simple question, is there an increase in cells in G1 phase in this activated population prior to differentiation into plasmablasts? And so we went back and we looked at this Fookie system again, and we looked at the activated B cells for the frequency of cells in G1 or S phase. You can see at 3 days after activation, there's almost equal frequency of cells in S and G1, but as over time, you could see an increased frequency of cells in G1 and a decreased frequency in cells in S phase, suggesting that these cells are potentially slowing down and spending more time in G1. And so what we then asked was, what can we basically use our understanding of what we found from genome organization and set up an experiment to ask that does prolonging cells in G1 phase actually promote increased blast differentiation. And for this, we used data that had been generated by Phil Hodgkin and Jay Zhu in their lab. And this experiment, what was done is cells were treated after activation with drugs that were able to prolong S or G1 phase. And then they were examined the number of cells which differentiated into plasmablasts over time. And what was found was that indeed, if you prolong cells in G1 with drug, you did indeed find an increased frequency of plasmablasts being produced as compared to cells in when the S phase was prolonged. And this is supported by recent studies from Hodgkin lab. But also, basically, from our point of view, was able to show that we could potentially, find a biologically significant findings from our genome-wide high-C analysis. And so, overall, what we've been able to show is that during B-cell activation, you have this large wave of transcription that occurs here and sort of at the end here, and this is. After this, we see genome organizational changes occurring in these 2 distinct waves. And what we found is that these changes in genome organization appear to be separated from the rapidly cycling cells. So basically, to conclude this story, what we found was that the genome organization occurred prior to the first division or prior to mitosis, and they previously thought that this kind of major architectural changes would occur after division, so that's quite a surprising finding. And we think these results are generalizable. I think this system has allowed us to ask these kind of questions which perhaps haven't been asked before. We know that, and interesting from the immunological perspective, the architecture is maintained as the population undergoes this expansion phase, this clonal expansion, and I think this makes a lot of sense. As you'd expect, if cell fate decisions are made prior to the division, you want to expand the population, you don't want to spend too much time changing the genome after that. And it also suggests that this rapid proliferation doesn't support the ability for the genome to change. And also, what we found is that the reorganisation and differentiation into plasmablasts is associated with prolonged G1, and we think that's quite compatible with literature. And so finally, I wanted to basically conclude about our lab and what we've done over the last 5 years. So we've used the strategy that I outlined at the beginning to identify a potential new target to treat allergic disease. And we're quite encouraged by this, and we're hoping that this will have potential going forward in clinic. We've actually established quite a few different techniques to study how the genome is organised in the immune system. We've made quite a few findings about how the genome is folded during the development of the immune system. And as Steve mentioned, what's allowed us to do is to collaborate with many labs, both within WeHi, nationally and internationally now. So that's been very, very exciting. And so our future plans, well, obviously there's many open questions in this area. As this kind of project this kind of work is in its infancy, and we've recently published this in this review with our ideas of the many different things that could be studied. One of the things we're quite interested in is how does a memory immune response work. We know that there's resting memory cells that are able to react more quickly to an infectious insult. And how does the genome look of those cells? How does it respond to this kind of rapid reactivation and things like that? But we're also mindful that we wanted to try and more meaningfully study what happens during disease. And so we're interested in studying blood cancers, autoimmunity, and what happens during aging. And what we're going to continue to do is to adopt or develop new techniques which will allow us to understand this. So looking at genome-wide DNA damage or DNA breaks by NSeq, working on how to capture interactions that are around SNPs in autoimmune disease. And we're going to continue to try to increase our ability to visualize either computationally or by using techniques such as chromosome paint. And so that leads me to my acknowledgements. I'm going to slowly go through the lab. I haven't actually thought about what I was going to say here. So, I mean, people know Tim. He's already done a Wednesday seminar before. He's done an incredible amount of work establishing all of the high-C data in the lab. And, it's basically a tour de force. His focus, his intensity is exactly what we needed to start a lab. And he really has even listened to what I asked him to do, which is quite amazing, and focused in on these specific problems. And everything basically on this second part of the talk is built upon his research. Christine has done a phenomenal job of bringing together all of our work in the asthma space, and I think that's going to continue on. And she's also starting to put her hand to studying how the genome is organised, which is very good. Luke has done an incredible amount of hardcore molecular biology, and he's done an amazing job of setting up all the CRISPR work in our lab, and that will continue on undoubtedly. And his work is this B-cell activation. He's the primary author on that. Hannah is the bioinformatics guru in our lab. She's done a huge amount she does all of our bioinformatics, in fact, and she's also co-supervised by Gordon Smythe. We continually hassling her to do more bioinformatic analysis for us, and she's doing a great job with that. And Nadia is the research assistant in our lab who basically keeps the lab running, who manages all of the mice and all of the annoying bureaucratic things that I ask her to do. Thank you very much. And also there's all the other members that have helped us a lot. Obviously, Stephen Nutt's been a fantastic supporter of our work and allowed us to be very creative and very supportive of all of our ideas. And other members of the Immunology, especially Phil and Jay, for contributing their data to our work. And other collaborators, Lynn Corcoran especially, she's probably not here, but she let me share her office for many years. I'm sure I annoyed her a lot. I don't think she listened to me most of the time, so that was okay. And Rima, thank you for being a great advocate for our research, especially in the asthma space. Inspiration has been the B-cell program. If it doesn't exist anymore, it's been inspiring as a scientific pursuit. Gordon Smythe, thank you so much, Gordon, for persisting with continuing to analyse our high-C data. It was quite difficult at the start. It was a difficult technique to master. And Gordon and Aaron wrote software in the absence of our actual data. So thank you very much for that. And eventually we came through with it. Thank you so much to people in Len Harrison and the people in Nizab, Nayar and Gaetano for providing samples and our continuing collaboration. Marnie and Doug for allowing us for a short time to be part of molecular medicine. The genomics wouldn't have been possible without Stephen Wilcox. That's been fantastic that he's been running this facility and allowed us to do our sequencing so rapidly and so easily. The bioservices, Tisha and Rhiannon, have been critical for our animals. And our collaborators, so Phil Hansbrough and his team in Newcastle, Philip Kalas for the Chrome 3D, Warren Alexander, Jane Visveda, Stuart Orkin and Sasha Tarakovsky have provided mice. And I'd also like to thank the finance people because I've annoyed them a lot and trying to understand how finance works. So Courtney and Lucy and Sally's been very helpful in fundraising, and communications, Aruni has always given us a lot of time. And I'd also like to thank my family, Allan, Arlo and Oscar, because they had to listen to me practice this talk and be very annoying to them. And also I'd like to thank all of the funders for funding our research. Thank you very much.
When you showed that there was a contrast between early differential and late chromatin,
yes.
your comment was that there's a structure that dictates and allows this expression.
Yes.
Why don't you conclude that the expression drives the genes?
Well, it can be both. I mean, I was more specifically focusing on what the genome organisation does and how that dictates transcription. So I think there is a pre-existing architecture that dictates the early transcriptional changes. But of course, it's true that the products of transcription then most likely will be involved in organising the genome, and there's no doubt about that. So we've thought about that, and people in the lab have talked about blocking transcription and seeing what happens, but that'll screw up the cells and all sorts of funny things will happen. We won't get into specific details. What did I want to do about that? Yeah, so this actually, that finding about the changes prior to the first division allows us to potentially look at the regulators of genome organisation like CTCF and RAD21 and other factors that people haven't been able to study because those factors are also important in mitosis. So we might actually be able to get an angle on that if we make a PROTAC or something to study some of these genes which are thought to be involved in genome organisation and study it prior to the first division.
Just curious, what was the drug or drugs you used to prolong G1?
I think it was, mimicin is one of the ones. No?
That wasn't one. That's it. I think it was progalanol.
What do they do?
It's a CDK inhibitor here.
Okay.
I think most of them are CDK inhibitors, I think. Yeah. Yeah, I mean, it's obviously those kind of experiments are not really perfect experiments. Obviously, there's many caveats, and they don't just block it in that stage. It was just kind of a proof of principle. It's a bit weak, I know that. So.
Okay, Andreas.
So you look at the architecture around transcription factor genes that are highly specifically expressed in either granulocytes or B cells or T cells. What about if you look at a transcription factor that actually plays a recognized role from knockout studies in 2 lineages, and then compare the architecture around that transcription factor gene between 2 different cell types? I guess PU1 would be a good example for that.
Yeah.
Have you done that?
No, we haven't done that at all. We haven't even looked at that. Actually, it's an interesting idea. Yeah, we'll have to do that in the future, I think. We haven't tried to do that at all. There's so many different things we could look at, so...
I was just wondering if the architecture could be the point of return from the differentiation standpoint.
Yeah.
You tried to reverse some of the differential changes before and after this.
And we haven't tried to reverse it. I guess people have done that in reprogramming, like as I showed before. So yes, you can reverse the architecture. I think that basically, as has been shown with reprogramming, you'll still have a signature of the terminally differentiated cell, but you can change the architecture back again, at least in the context of reprogramming. I know that that's true, but we haven't done it ourselves.
Rhys, you mentioned early on that gene-gene interactions are cis rather than trans.
Yes.
Most of them. Does that allow you to think about comparative genomics to get to think about why certain species keep genes on certain chromosomes? Does that provide any insights into how things might work and as to why some species have a lot more chromosomes than other? I mean, is it a stupid question? Or is
It's not probably not stupid, but I haven't thought much about that. That's
I mean, that's just or is it just...
Yeah, no, that's definitely valid. I mean, people are using high C in all different settings to study these kind of things.
I just wonder, because, you know, you've
about
got...
conservation of regions or something like that,
You've got this wealth of information
Yeah.
of various species
Yeah.
and how many chromosomes and what genes
Yeah,
are
sure.
on what chromosomes,
Yeah.
and whether that can help you thin out what the key mechanisms are
Yes.
in driving what are fairly conserved immune responses.
Yes, yeah. Well, we have found that some of the regulatory regions that we've identified are conserved, but we only really concern human and mouse, so we haven't looked further than that. But that's definitely a good idea.
So, I guess most people were originally surprised by the sheer number of DNA binding transcription factor binding sites.
Yes.
So, are you able to use this data to sort out which ones are kind of real or functional binding sites and which ones are maybe just there and not really interacting?
Probably not at this point because the resolution of the high C data isn't high enough to get right in and pinpoint what's happening in specific binding sites. I'm not sure we can do that at this point.
I think it's fair to say that more regulate loops than regulate transcription.
Probably very naive, but related to Melissa's question, if you would reverse the architecture, do you think you would reverse the timing to the first division? Would you then delay the timing again?
You reverse the architecture. I'm not sure. Reverse. So, no, I don't know. I'm not sure. I haven't thought about it.
Charlotte first.
So you talked about perhaps it's generalisable that chromatin architecture changes before the first division.
Yes.
That may be the case for something that's in a quiescent state that gets activated.
Yes, yeah.
But what about something that's in flux continuously, like gut tissue or hematopoiesis
Yes.
or something?
I don't know
what does
that.
the chromatin architecture potentially look
Yeah.
like there? More subtle, more gradual?
Yeah, we haven't tried to study that actually, so I don't know if that's the case. It's a good question actually. Whether they're when they're already cycling and they change. I mean, it depends. I guess what our work is trying to say is that if they're cycling rapidly, they're unlikely to change their genome and they will slow down. I guess that's the idea that we were trying to say about that second wave. So in that context, the cells might actually slow and then change. That was the idea. That is the case in some stem cell culture systems, but I'm not sure. We haven't studied that in detail.
Just sort of a more general question. When you look at all the long range interactions that YC is detecting, and in your B cell example during this sort of 30-hour delay, there's a lot of these long range interactions being broken and new ones being made. Sort of, what does that look like physically? What's directing those? How do they find each other? What sort of structural and support factors are involved?
Well, I think it's I think it's probably, as we suggested earlier, like transcription factors are probably the differential binding of those would somehow physically alter, I guess, the chromatin fibre in some way, I guess, and or allow other factors to form a complex. There's all this information now about phase separation. So there's thought to be, create this phase-separated compartment. If you have a certain density of binding of factors, that they'll come together and then the enhancer promoter will interact within that phase-separated compartment. But I know there's not much information about that at the moment.
I wonder whether you see any of the nucleosome structure in your high C maps. Are they repeating long-range interactions?
The nucleosome structure, we're probably not. We're not able to get that with the resolution that we're looking at the moment. We're really looking in like 20 kilobase spins, so we're not able to get that nucleosome level resolution. It probably is more appropriate for a TAC-seq or something like that.
The non-coding RNA you may
Yes.
have identified upstream of GATA3,
Yes.
does it have a strong enough effect that it may cause the same sort of disease that mutations in GATA3 cause?
Well, we don't know yet. Yeah, well, that's what I was trying to get at. We're trying to sort of work out the functionality of that gene, if it's important or not. We're going to try and overexpress it in B cells, for example, and see whether it can drive GATA3 expression in that context. I guess ideally, it would be amazing if you could show that we could target that and alter GATA3 expression. That would be the ideal situation. But yeah, I don't know if there's probably multiple genes that regulate that fate, and whether this one is just one small part of that, it's hard to know at this point.
Okay, we'll pack that up. Thank you.